Our next talk is by Pandam Salifu from KNUST, Ghana. He's going to be talking about collections in Africa, an overview of what exists and the possibility of a microbiota collection. Pandam, please go ahead and share your screen. We are ready for you.
Thank you for giving me the opportunity to present this afternoon on what exists in Africa and the possibility of collection microbiota. As the chair has introduced me, I'm from Nkrumah University of Science and Technology. Today's presentation, I would focus on three aspects. And the first, what exists, and then possibilities, what are available for collection, and also the facility and resources that are available across Africa, and specifically narrowing down to Ghana as well. Over the years, I have had the privilege to manage one of the cutting-edge research centers in Ghana, and being responsible to manage various research projects in collecting samples and across, and also to keeping storage for the samples, and of course, being able to organize and ship this particular sample to collaborators across the world. And so we have heard a lot from the previous presenters on various collection of microbiota research that has been done from food and other environment, and of course from humans. And so in Africa, in terms of collection of microbiota, there has been a limited work in microbiota, even microbiome of recently in human microbiome research has been conducted. So to give you a guess, about 168 human microbiome studies has been conducted. And about 114 of this published research work were conducted in the last 5 years. And even if you are aware of African continent, there are about 56 countries and only 33 countries in which microbiome related work has been conducted. There are many countries and where limited work has been done in terms of microbiome and also microbiota collections. So this was a review that was conducted by Alali and co. So in terms of the microbiome research on the continent, majority of the body sites that were collected to undertake this work, the number of samples that were conducted are less than, are between 5 and 10. And the body size include the brain, breast milk, eyes, gut, lungs, and other part of the body were undertaken. And these are the various countries that were being, that participated in the research. As you can see here, there are still countries such as Tunisia and that hasn't been, there is no report of microbiome that has been conducted in those countries. And so the body size in terms of percentage, I mean, this diagram illustrates some of the sites, the number of or percentage of work microbiome research that has been conducted. But majority, they weren't much of this work were done in collaboration with Northern partners. So the sample distribution of microbiome research in terms of age were fairly concentrated on adults and also children. And so many of the work had sample size less than 1,000 participants. And sex distribution, this is fairly across among the researches that were conducted. Majority of the studies that were conducted were between 500 and 1,000 sample size. And South Africa and Kenya were some of the countries in which much of the microbiome related and microbiota research has been conducted at large. And so in terms of disease focus, most of the work focused on HIV-related, non-communicable diseases, and of course, malnutrition, and also bacterial vaginosis. And so over for the past 9 years, H3Africa which consists of many research groups across Africa, came together, that was 10 years ago, to initiate and really span in terms of genomics research in Africa. And this group, the consortium, where they have been supported by NIH and Wellcome Trust. And so many of the research collects are engaged on sample collections, but they are mainly focused on humans. And so for research projects, about less than 30 of the projects were research, but also had collaborative centers, biorepository centers that were funded to collect samples and to be able to share with researchers across the world. And there is one coordination center that coordinates all this project. And of course, sample collections and all those kind of things require ethics, legal, and social implications. Centers were funded to able to support the various research groups in terms of ethics and social implications of genomics research, and also to conduct research on African continent regarding social implications and the legal framework. And majority of these projects are also supported by bioinformatics network, H3 Bionet, for which I am a co-PI on this particular project. Then in the first round, most of the projects were funded 5 years, and that was between 2012 to 2017. And subsequently, new rounds of projects were funded. And so there was also the need to see that training in bioinformatics relate sustainability aspect of training bioinformatics. About 4 projects were groups were also funded and to undertake bioinformatics training in Africa, which is still a new part of it. Fortunately, I was my group was funded to also undertake the bioinformatics training, for which we started conducting bioinformatics training for the past 2 years. And so in microbial research related in this consortium, 2 centers were funded one is focusing on respiratory microbiota of African children, and the second center mainly focus on microbiome and genome research. And so 4 projects were funded in focusing on microbial research. And so genome characterization and surveillance of microbial threats in West Africa was one of them, and nasopharyngeal microbial and respiratory diseases in African children. It is interesting to note that in West Africa, very few, we had 2 research that was also funded to collect this. In terms of the biobanks for collecting samples to support the research work in Africa, and as the biobanks were supposed to collect some various researchers to send their samples to these biobanks and to archive and make them freely available to researchers who request and want to do additional analysis. And so 3 were funded. In addition, there was additional site that was funded to be able to develop biobank solutions or support, such as open source laboratory management and tools, so that which can support sampling and support archiving and tracking the samples. And so the 3 biobanks, one is located in South Africa at University of Witwatersrand, and also had, which was led by Elizabeth Mine. In Uganda, Moses Juloba, who also were funded to establish integrated biorepository of Africa, H3Africa, and in Nigeria, VARA Research Institute was also funded to archive samples that are collected. And so they made this centers, the biobanks, they accept samples across Africa. So, they are just to archive them and make them available for researchers. And among the 2 and the 3 biobanks, the Uganda Biobank, their catalog is freely available online, and there is a form if one want to request samples from their catalog, and there is procedures as how you can place samples. There are over 40 different projects and samples that has been collected and stored.
Oh, Pandam, please, you should be rounding up. Thank you.
Okay. So there are at the Uganda biospecimen storage, there are various samples that has been collected as listed here. And so this is the site from Uganda biorepository catalog. At the site, you'll be able to place samples. In terms of the Nigerian VARA biobank, I mean, they have indicated they have various samples that has been stored for sharing with other researchers, but they don't have it online readily available for submission of requests. So apart from that, there are also various biobanks that have been established by various researchers and research group. One is the Trypanogen, but they've mainly focused on human, collecting blood plasma and whole blood assay. And there is also a Syrian project that investigates through research, and mainly the target is to recruit 3,000 cases and control, and they collect blood, serum, and those kind of things. According to them, they have been able to collect over 9,000 samples that are available for collection. So for possibility of microbiota collections, it's enormous. Africa is a big continent with many countries, many wildlife, different race, ethnics. So there is possibility to collect in many parts of Africa, as indicated. Of course, there is also a possibility to collect even in animals, the food, both wild and domesticated animals, the environment, and those kind of things. So for facilities, I'll just present you one of the facilities that we have in Ghana, for which Kumasi Center for Collaborative Research, which I happen to have been the head of laboratories and manage most of the sample collections. KCCR, for instance, has many cold storage facilities, both minus 80 degrees and over 180 degree freezers, and more than 30 deep freezers available for sample collection. We have on site also liquid nitrogen generation facilities and dry sippers capable of, and so there is capacity to undertake sample collection from any part of Ghana and neighboring countries. I've run out of time, and I would like to acknowledge support from NIH for two of my projects. There was separate project for training, and also the H3 Biobank project. I would be available if you need any questions, please forward it.
Thank you so much, Pandam, for such a great talk and for throwing the light on the state of microbiome collection in Africa. Because of time, I'll just read one question for you to answer, and then you can answer the rest in the Q&A. Mary would like to know, he said, I would like to have a little knowledge about the countries that are gray in terms of microbiota research. Is it that research is being done and not documented, or not done at all?
There is the information that are publicly available. To my knowledge, indicates little or nothing has been done. And those that have done work and made their data available that have published, but talked about. But there is also one component to understand. Most of the countries where this work were conducted was done in collaboration with researchers outside Africa.
Okay, thank you so much.